In this part of the video, we are going to solve the problem A from the section B of the problem 4, part B, problem 4A, which this mentions. Use the procedure of part A to decode these messages. Remember transcribed messages DNA codon to mRNA, and then translate codons into amino acids. And this problem says TGA, CGA, TTT, CTC, ACT, ACA, CGC, GCG, CTT. So in order to solve this problem, we are going to go back to look at the part A of this questionnaire and search for ilustración A, mapeo de codones ARN a aminoácidos. So we can give us an idea about how we can translate the code of the problem into the RNA amino acids.
Well, the part B of the questionnaire involves the transferation of DNA to messenger RNA. This involves the 4 bases that conform DNA that are adenine, thymine, guanine, and cytosine. Therefore, being exchanged adenine into thymine, that thymine will transform into uracil, and cytosine into guanine. Well, they are arranged in a 3 group of letters that are called codons that transfer in order to anticodons to bond with them that have amino acids. These amino acids mainly start in the change with the amino acid methionine, and then continues to form a larger change of amino acids. You will appreciate that in some examples or some exercise, they will not start with the methionine. This implies that this change have already been started. Then there will be found that some are instruction for stop, that will be mainly UAA, UAG, and UGA. This will imply that the change have already broken and then is not necessary more proteins to be formed, or a change will bond to another change to produce even better or bigger amino acids or proteins. Well, this mainly will transfer into another parts of the cell in order to fully develop as proteins, which then leave the cell membrane.
I'm going to continue with the part A, which corresponds to the procedure to get the mRNA from the DNA code. And well, in the first question, they ask us to get the mRNA codons from the DNA code that they give us. And well, as we should know, to get the mRNA codons, we need to make the transcription of the DNA code to the mRNA. And for example, in the question they give us the code TAC from the DNA, and to transfer it into mRNA, we need to change it to AUG, where we are changing the T with the A with the U, and the C with the G to complete this process of the transcription. And then the second question asks us to get the amino acids that corresponds to the codons that we got in the first question. And for this they give us a chart with all the amino acids that exist, and to complete this we just need to get a codon and use the chart to get the correct amino acids following the letters that correspond.
DNA is a huge information database that carries a complete set of instructions for making all the proteins a cell will ever need. Although there are only 4 different bases in DNA, A, C, G, and T. The order in which the bases occur determines the information to make a protein, just like the 26 letters of the alphabet combine to form words and sentences. DNA is transcribed or rewritten into RNA in a very complicated process called transcription. Think of a transcriptionist that takes spoken words in one source, like a courgette, and makes a copy of those words on paper. During translation, the mRNA transported to the cytoplasm is decoded or translated to produce order of amino acids in a protein. Translation requires numerous enzymes, that's why it's very difficult to do.